Hello everyone, my name is Katerin and my narrated PowerPoint presentation will be on prenatal development. Prenatal development is a development of a baby from a single cell within a woman's body. This development is split into three major stages: germinal, embryonic, and fetal. The first stage is the germinal stage, which begins at the point of conception when a sperm fertilizes an egg within one of the fallopian tubes. This stage lasts for 2 weeks, where the fertilized egg or zygote travels down the fallopian tube towards the uterus. About 24 to 36 hours after conception, rapid cell division begins and the cells slowly separate into two masses, one of which later forms into the placenta, while the other later develops into the embryo. The rapidly dividing cells eventually develop into what is known as a blastocyst that consists of three layers: the ectoderm that becomes the skin and the nervous system, the endoderm that becomes the digestive and respiratory systems, and the mesoderm that becomes the muscle and skeletal systems. When the blastocyst reaches the uterus, it implants itself onto the uterine wall, and at that point it begins the embryonic stage and becomes an embryo. The embryonic stage, which begins at week 3 and ends at week 8, plays a vital role in the development of the brain, since it begins upon the basic structures of the brain and establishment of the central nervous system. This stage also marks the development of facial features, limbs, all of the major organs except the sex organs, and the heart officially begins to pulse. The beginning of week 9 starts the fetal stage, which continues up until birth. At this point, the embryo is now a fetus, and the body systems and structures of the embryonic stage continues to develop. Reflexes begin within this stage, and all of the organs and parts of the body become fully developed. This stage is the longest in duration and concludes the first trimester and leads into the second and third trimester as well. All in all, prenatal development is marked as the time of remarkable change that helps set the stage for future psychological development. There are various external dangers that can pose a threat to a developing fetus. The first type of threat are genetic problems, which are diseases caused by the change in one's DNA. Down syndrome is a result of an extra 21 chromosome and is considered one of the most common genetic problems that can affect about 1 in every 1,000 infants. Inherited diseases occur when one or both parents carry the gene that causes the illness. Sickle cell anemia, cystic fibrosis, and Tay-Sachs disease are all examples of this type of disease. Sex chromosome problems affect infants when there is a loss, damage, or addition of either one or both sex chromosomes. Examples of these are Klinefelter's syndrome, which is an extra X chromosome, and Turner syndrome, which is a single X chromosome. Another type of threat to prenatal development are environmental problems. Harmful environmental elements that can affect the fetus are known as teratogens. Examples of these are the consumption of alcohol, the use of cigarettes and drugs, and even the pollutants found within the air. To further explain the effects of teratogens, I found 2 research articles that further studied the effects caused by the consumption of alcohol and the presence of nicotine. My first article was the effects of prenatal alcohol consumption on cognitive development and ADHD-related behavior in primary school age, a multi-level study based on meconium ethylglucuronide researched by Anna Eichler, Linda Hudler, Julianne Grunitz, Jennifer Grimm, Eva Rabe, Tammy W. Goki, Peter A. Fashioning, Matthias W. Beckman, Oliver Kratz, Gunther H. Moll, Johannes Kornhumber, and Hartmut Heinrich. According to the article, prenatal alcohol consumption is one of the most validated prenatal risk factors that impairs a child's development, and yet the use of alcohol during pregnancy is still at 20% in the United States. Exposing the fetus to alcohol can lead to various developmental issues such as fetal alcohol syndrome, an irreversible syndrome that causes brain damage and growth problems. It also causes cognitive and attentional deficits, as well as symptoms of hyperactivity and impulsivity. While there have been many studies to test for and detect the issues pertinent to alcohol consumption, this was the first study to observe a biomarker or a traceable substance that is introduced into an organism called ethyl glucuronide or ETG. ETG is a minor ethanol metabolite that begins to accumulate in the fetal gut after the 20th week of gestation, all the way until birth, and can be detected within the meconium of the newborn. The assessment was conducted at two varying times, between the years of 2005 to 2007, and also between the years of 2012 to 2015. The subjects of the study were 18 years or older women that were in their third trimester with 30 weeks of gestational time. During the first assessment time, the mothers provided a self-report of their prenatal alcohol consumption during their third trimester. 1 gram samples of meconium was collected from a total of 88 infants within the first 2 to 24 hours after their birth. Among the samples, 44 newborns were recorded as a control group by having no exposure to alcohol, while the other 44 newborns experienced exposure to alcohol. To study the effects varied by dosage, 22 infants that were exposed to alcohol were documented at just over the detectional limit of 10 nanograms per gram. However, the remaining 22 infants were exposed to levels greater than 154 nanograms per gram, which is among the 50th percentile measurement of alcohol consumption. The second assessment time was recorded when the children reached primary school age. Cognitive development was measured using the Standardized Intelligence and Developmental Scales. ADHD-related behavior was measured by the mothers using the German ADHD rating scale, while ADHD-related aspects such as event-related potential and performance data were recorded using cued go and no-go tasks. According to the results of the study, children with a meconium ETG that was above the detection threshold of 10 nanograms per gram showed reduced P3 amplitudes in the ADHD-related event-related potential and performance data, which is reflective on the allocation of attentional resources and executive response control. This can be interpreted in the sense that prenatal alcohol exposure can lead to specific attentional impairments. Also, the children that showed a meconium ETG that was above the 15th percentile of alcohol consumption, which was greater than 154 nanograms per gram, scored 6 points lower on their IQ scores, which shows a direct impairment to their cognitive performance. Lastly, when comparing the children that were exposed just beyond the detection threshold of ETG to those within the 50th percentile of exposure, the results showed a clear indication that greater ETG values was directly correlated to higher ADHD-related behavior. My next article is the effects of prenatal nicotine exposure on infant language development, a cohort follow-up study researched by Carmen Hernandez Martinez, Nuria Voltas Moreso, Blanca Ribot Serra, Victoria Arrija Val, Joaquin Escribano Macias, and Josefa Canals Sanz. According to the article, tobacco is the most widely used toxic substance during pregnancy. 11 to 30% of women either smoke or are exposed to secondhand smoke during their pregnancy. However, this statistic rises to about 50% when considering individuals from poor, young, and urban populations. There have been countless amounts of studies done on the effects of nicotine on pregnant women and their offspring, all of which resulted in various health problems as well as behavior and cognitive impairments. Exposure to this toxin varies in its potential damage and has been related to increased irritability and excitability when infants, poor motor and cognitive development when toddlers, and even low performance in reading, language, and verbal learning well into their primary school years. This study adds new longitudinal data from 6, 12, and 30 months old that considers the comparative effects of smoking and secondhand smoke exposure on an infant's cognitive development and their language development. The sample for this research consisted of 158 women that were all recruited for the study when they were pregnant at no more than 11 weeks of gestation in their first trimester. The women all had to be over 18 years of age and healthy with no sign of chronic illnesses. Their babies also had to fit the qualifications of being full term with a normal birth weight and no medical problems exhibited their first days of life. A survey was administered at each trimester to determine social demographic along with information regarding maternal and familial smoking habits. During the postnatal follow ups at 6, 12, and 30 months, cognitive, language, and vocabulary development was surveyed. At all three postnatal follow ups, the Bailey scales of infant development was used to measure cognitive development. Language development was collected using the MacArthur Bates Communicative Development Inventories at the 12-month postnatal follow up. The Peabody Picture Vocabulary Test was given at 30 months. Concluding this 3-year follow up, it was found that maternal smoking during pregnancy and secondhand smoke were associated with poor cognitive development. Even low amounts of exposure were related to short and long term deficits. According to the researchers, language development was mostly consistently affected, which can particularly lead to spelling, reading, verbal learning, and receptive language problems. In conclusion, using meconium ETG levels received the acknowledgement for its capabilities to predict a child's development for the very first time. The biomarker was able to suggest a relationship between prenatal alcohol exposure and a child's cognitive development as well as their symptoms of ADHD related behavior. Nicotine was shown to be a direct contributor to short term and long term language development, along with cognitive deficits as well. From the early stages of prenatal development, the baby is given oxygen and nutrients through the bloodstream of the mother. The placenta and the umbilical cord unite the mother and the fetus, which allows the exchange of necessary growth factors. Since the mother and the growing fetus are directly united, the exposure to alcohol and nicotine is directly reaching and harming the unborn child. As stated before, prenatal development is marked as a time of remarkable change that helps set the stage for future psychological development. Without correctly caring for the fetus while it is growing within the womb, many psychological developments can be affected and can make it difficult for the child to reach and conquer developmental landmarks as well. For instance, one of the very first stages in the development of a newborn is the sensory motor stage. From birth to 2 years old, infants rely on their motor skills and senses to understand the world around them. With correct development of this stage, children will then lead into the preoperational stage that begins to push toddlers between the ages of 3 to 7 to use words and numbers to make sense of their world. However, exposure to nicotine and alcohol is known to impair cognitive development and even delay motor skills. By crippling one stage of development, the next stages become even harder for the child to achieve, thus delaying their psychological growth. I believe that it is important for individuals to understand the true harms behind the exposure of these teratogens to the development of a baby. Many women still overlook the dangers that drinking alcohol and smoking cigarettes can cause to a growing fetus. But after learning the results of these studies, there should be a greater effort worldwide to to end all alcohol and nicotine consumption while pregnant. Every child deserves to grow in a developmentally sound environment, and their very first environment is within the womb. So, further banning these toxins from an individual's life will not only benefit them, but the baby in many more ways than one.